Implantation of a prosthesis involves complex surgery conducted in a sterile environment. Still, sometimes implants get infected with dangerous bacteria. The consequences can be very serious. Why does this happen, and what can be done to prevent it? Angel from Barcelona had his knee joint replaced with a metallic prosthesis. At first, it seemed the routine operation went well, but complications quickly became obvious.
Al tercer día o al cuarto día, esta rodilla no acababa de...
3 to 4 days after the surgery, the knee didn't improve. It got worse. Bad inflammation, pus, black spots around the wound. The doctors found it got infected during the implantation of the prosthesis.
The infection endangered the whole limb and could have become life-threatening. To stop it from spreading, Angel had to have intensive antibiotic treatment and several more operations. His knee has never fully recovered.
What makes such infections so difficult to prevent and to treat?
The main problem is that bacteria contaminating the surgical wound attach themselves to the metallic implant and grow into a biofilm, a matrix that wraps them up and protects them from the immune system, as well as from the action of antibiotics that we're administering. And the only solution in many cases is to remove the infected implant, which greatly complicates the situation for the patient.
Infections occur in 1 to 3% of surgery like this. One possible solution could be to find new antibiotic molecules that combat drug-resistant pathogens and then coat metallic implants with these molecules. Researchers in this Barcelona laboratory are screening thousands of microscopic algae species looking for compounds that can destroy biofilms.
Microalgae produce large quantities of secondary metabolites with functions that are largely unknown. The algae survive in aquatic environments which are full of predators, so they need protection. We think these secondary metabolites can have antibacterial properties.
Finding new antibiotics is just one of many challenges tackled by this European research project. Scientists, together with manufacturers of prosthetic devices, are developing a set of necessary methods to produce and test infection-resistant implants.
Once we have these molecules, we'll test them against most common pathogens. We'll analyze their safety, and only then we'll be able to find the best ways to apply them on implants using nanotechnology and surface coating.
The pig's immune system is similar to humans, which allows for safety tests of new implants for future human trials. For this test, researchers in Copenhagen placed small metallic implants into a pig's leg bone and analyzed the results several days later.
Professor, how do you test these materials on animals?
We do recognize the drill hole in which the implant was inserted together with the bacteria. And what we are doing is to test different coatings of these implants in order to find out if they can prevent the infection in spreading out into the bone tissue.
Researchers estimate they will need several more years of research before human trials of infection-resistant implants become possible.